Yeah, we are back. We already learned mass-univariate regression ERPs, including continuous effects and marginal effects. And I think this is all really advanced stuff already. And I think you should all be proud that you're still sticking around, right? But we will continue. There's no time to stop this train. We will only stop after overlap correction, and we will just jump right into it. And we will start next with making the straight lines that we had so far, these straight regression lines, bent a little bit, right? Okay, let's imagine you have a scenario again where we look at a single time point, because it's always easier to think of single time points. And you have some kind of predictor value, let's say saccade amplitude or brightness, contrast, whatever you fancy, depending on which field you come from. And this is the signal to be explained, so that could be the P100, for instance. Now, the underlying individual trials, as you can see here, they are not on a straight line, right? They have this kind of maybe logarithmic shape, or they could have any other shape. But in this example, there's not a linear relationship. You can fit in a line, but then, I mean, it wouldn't make much sense for some of these values. In worst case, imagine you have a quadratic relation, then the line would be flat, even though there would be quite a lot of relationship to the saccade amplitude. So wouldn't it be nice if we could allow this line to bend a little bit, allow this line to have different values? And one thing that people are doing is that they say, okay, maybe let's just bin the variable. So we're going to define one predictor that only models this trials, one predictor that models this trials, one predictor that models this trials, one predictor models these trials, etc. And then each of these will have a different beta, different coefficients. It's just 0 or 1. You're either in that group or in one of the other ones. And by doing something like this, so here we have predictors. We could sum up, and while there's now only one predictor available, and the other ones are all 0 everywhere, of course. So the sum, bear with me why we are summing here. But we're always summing. We have this matrix products, etc. But we can sum up and we can predict the signal, and suddenly we are closer, in some sense, to this underlying nonlinearity, right? So we moved from fitting one parameter here to fitting 1, 2, 3, 4, 5, 6 parameters here by making this by categorizing basically a continuous predictor. Now, the issue, of course, is that in biological signals, it's extremely rare to have this kind of step functions where you have a complete change of the variable. It would be nice if we could do something similar that we have here, but have like neighboring, like have a little bit of overlap between these categories, right? So that would enforce that we have a certain type of smoothness in our fit. And that's exactly what we're going to do. And this is called spline regression. So we're using, instead of using this like 0 and 1 boxcar basis functions, what we are using is a spline set. And there are many different ones. We are using the so-called B-spline set here for various reasons. Yeah, okay. I don't want to get too technical here. And as you can see, you have now different basis functions. They're non-symmetric and they are importantly overlapping with each other. So in order to predict the signal that we have here, you would need to sum all of these here together. What you're seeing here is actually already one step advanced, and you will look into this in more detail in the exercise, because this is already the basis function weighted by the model fit. So already weighted by the beta that you had, that you needed in order to get this curve here, right? The same, this was the same here. The actual basis set all have the same height, and then you scale it by the predictor you're estimating, by the betas you get. And if you do that, then hey, this is great. We can get any more or less any nonlinear shape. Yeah. For those people who already know over and underfit, I will discuss that in a second. How does this now integrate in our general model of Unfold idea kind of model? Right, what we need to adapt is the design matrix. Instead of having one predictor for the continuous effect, we have to split this up into this basis set, right? So this looks like something like this. So instead of one, we now have, in this case here, 5 new predictors. And they will have certain value. Depending on this value that you have here, they will also have a certain value. So basically, you kind of look up what kind of what kind of overlap do I have? Where am I, and which of my basis functions is active as much? And if you then fit it, of course, then you will get also many more predictors here. And these predictors generally are not straightforwardly interpretable, right? You can look at one and then you say, okay, this one has a little bit of activity. It's somewhere in the middle. Probably in the middle, there's a little bit of activity. But because the neighboring ones can also always modify this behavior, this is not straightforward. And you guessed maybe you need marginal effects. Or one easy way to do it is to use marginal effects. And if you come from the MATLAB Unfold toolbox, there are not many, but I mentioned it anyway. This is kind of what we implicitly implemented there, also with the UF at marginal, UF predict continuous combination, right? But back then we didn't know better that this all called marginal effects, and in Julia version, it's a little bit more conceptually straightforward. We can look at this also in a different way. So this would be a design matrix. So you have the events here, or the trials, the repetitions. You would have one predictor that's just one everywhere. And then maybe this is a linear predictor, right? You see the linear formula here. And if you have a spline set, then and this is sorted by the spline set, right? You have the very large saccades, and here you have the very small saccades. And you can appreciate that this is some kind of basis function just now flip to the side. So you look on top of the basis function, basically. Right, just a design matrix in the wild, basically. Okay, let's give you an example of why this is important. And here, Olaf, this is a study. He did many years ago, and we basically analyzed it with this tool, and it's published here in Journal of Vision. It's a nice paper. Check it out. No, it was fun to write, and what the task was here was people were presented with visual natural scenes, and they were just looking around. I think at some point a target appeared and they had to press a button when that happened or so. But the task is not so important. They just explored the stimulus more or less freely, right? And each of this fixation, I mentioned this before, we could take now each of this fixation and model it as one event, basically, in our data set. These events were modeled using two predictors. One is a spline for saccade amplitude. So this is also the way you would put this in. So instead of just writing saccade amplitude, you have to tell the tool, oh yeah, expand this to a larger set of predictors. And we are using spline here. You could also use, I think, you could use, for example, also a polynomial expansion or something like that for people who know what it is. But splines have better properties and generally are in favor compared to a polynomial expansion. Anyway, we are focusing on the saccadic amplitude. I will talk very briefly about the circular spline on the next slide. So if we do this and we calculate the marginal means, the marginal effects, this is what you already did in the last exercise. This would be kind of what we got for an channel OZ. And what you can see here is that if we kind of make the distances of where we evaluate the function similar, so this is a step of 5, a step of 5, a step of 4.5, you can see that the distances in the data here are not linear. Here, actually, they are linear. So at around 170 milliseconds, this is roughly linear. But here it's clearly nonlinear. And here again, it's clearly nonlinear, but the reverse pattern that we have here. So, and I find that really fascinating and important that we can also over time then have the change from a continuous to a nonlinear to another nonlinear effect. It's maybe not surprising in hindsight, but I think it is absolutely important. And we were not the first one to show this effect here. Other people have shown it before as well. So that seems to be something that's quite well replicable. Now, I just want to show you a different, maybe then the connection gets even clearer, a different visualization of this effect here in the gray area, right? So we average over this time point, and we put now basically the color we put on the x-axis, so between 0.6 and 15, and we put the amplitude on the y-axis, and then you can see here, this is this nonlinearity that you can also visually see here as well, but now maybe in a more linear regression kind of style that you would typically look at it. It's of course hard to do because showing this plot for every time point is impossible. Okay, so at least in this paradigm, we clearly see nonlinear effects, and there are many of them, and they look quite nice. There are some individual fits, model fits. Exactly. I promise you also to talk about the second bit, and the second bit is a circular spline. So we also looked at the, you can make eye movements from, let's say, from the center to the top, to the right, to the bottom, or to the left, or from any point, actually. They have an orientation, right? And so we just put the orientation as another predictor into the model. And now, of course, orientations are circular, right? So there's no difference or little difference between 359 degree and 1 degree. There's just a 2-degree difference. But if you would think of it as a linear effect, then they would be at opposite ends. So somehow we have to bend this x-axis basically around. And that's also possible with circular spines. I don't show the basis set here, but yeah, this is also implemented. And as you can see here, we do see this kind of circular effects also here at lateral electrodes. And if you make a saccade to the right, then the left visual cortex is more active. And if you make a saccade to the left, the right visual cortex is more active. And for up and down, it's a little bit more difficult to see. This is, of course, interpreting now this kind of 3 topo plots. We didn't do source localization or something like that. But you can equally well do more fancy things. And of course, you could also do your own basis set and various, yeah. Also, yeah, other customized things, but this would be a topic for a different workshop. I mentioned before that right now, or if you notice here, we are always defining how many splines we have. And it could be that you put this number to a really, really high number. And then basically you have for each event, you have your own basis function. And then you get something that's called overfit. So each of the line is overfitting the noise, basically. Or you could have too few splines. So you try to model some kind of complex nonlinearity, but you have only available to maybe model a simple quadratic effect or something like that. And then you would get maybe out a linear effect, even though it should be something in between these two. And the really big and unfortunately unsolved question to some degree is how to choose this number of splines. The way we are right now experiencing our models, etc., is that if you stay below 10, and even I typically just take 5 or 4 splines, that seems to be right. I'd rather underfit a little bit than overfit a little bit. The good news is there is some kind of regularization over subjects because you're fitting this model for each individual subject, and then you're going to average the marginal effects, for instance, over subjects, and that will regularize a lot of the overfits that you're getting. So there's some, yeah, we simply don't know, and of course it also depends on the quality of your data, the amount of your data, and well, simply the shape of your relationship, right? I do think that why do I then recommend to use like 5 splines or something like this? Because I think most of the relationships we have are not that you wouldn't expect to have a really crazy relationship in most of the data, or maybe you should, but yeah, we are coming from a world where we're not even fitting linear models, so linear relationships. So maybe we can slowly open up to more complex models. So this is the point. There are automatic solutions trying to address this issue. One is basically on cross-validation. This is already implemented in MATLAB Unfold. It is in principle implemented in Julia. We just have to write the tutorials, etc. As well. It's not particularly difficult. Actually, just some days ago, I asked a large language model to implement this for me, just because I want to see whether the large language model can do Unfold. Turns out they can, and they actually implement it correctly. So it's not a rocket science, but we just haven't come around to do it yet. The other prettier solution is to use something called penalized splines via linear mixed models. And this is also one of the reasons why I went to Julia in the first place, right? To use this linear mixed models, because I knew you could just use many basis functions and then treat them as if they are a random effect. This is now complex, just for people who know what I'm talking about. Treat them as a random effects and automatically estimate how much shrinkage there should be of the coefficients. So there's like, it's a type of penalization for large beta coefficients for any of these splines. And there's a kind of a tutorial. We started with it, but we're still in the mess around stage to get this right. But at some point, this will be there. And then you don't have this issue anymore that you manually have to choose the number of splines. Right? Okay. One step at a time. If somebody is super interested and motivated, I'm totally happy to collaborate on this and get this running. I have a notebook that's half ready, but the details are always tricky. Yeah, I'm very happy to support this. Okay. Well, how to do this in Unfold? That's fortunately extremely simple. You basically the only thing you need to do is you need to add that you're using this package bspline kit, because this is where the basis functions are created. And if you have Unfold loaded and bspline kit loaded, then Unfold knows, oh, there's an extra package. I'm gonna how do you say, unlock new extra functionalities. And then you can use in the formulas, you can use the spline kind of command, and Unfold does the rest for you. And if you use marginal effects, then this will just out of the box just work. If you plot ERP the coefficients, then you will get this, what I said before on this slide. Let me briefly show it. Then you will get this a little bit uninterpretable mess here of coefficients. You will see. You can just try it out if you're interested in it. And that's so much theory for that block. I think we already covered a lot of theory, so I try to keep it a little bit short in general. Happy to take questions. And okay, so there's a question. Does this mean a column in the matrix corresponds to a spline? Wait, I guess this is a question to this slide here. Yeah, I think this is just a terminology question, what you call a spline, whether you call the whole basis set a spline, which I think is typically done, or whether you call one of these things one of the spline functions. So I would say this is one spline function. Exactly, it corresponds exactly to that. So what we had here, one column basically corresponds to one of these. But of course, they are not sampled in this nice iterative way, but they are sampled in wherever this trial has a value, right? Yeah. Thanks for the question. The question is whether I'm aware of any other nonlinear effects that Unfold helped to uncover so far. For sure. So we have René wrote a nice paper on modeling duration effects as well. So you can imagine like duration, reaction times. There are many things in the brain that have a variable duration. And we are just treating it as if any trial has the same brain activity, regardless whether people were answering fast or slow, or whether looking away quickly or looking away slowly. And he has a nice paper. It's a preprint currently under review in Imaging Neuroscience. And yeah, we basically show that this is possible to model as well, and we do find effects of that. Also in fMRI, by the way, for reaction time. Yeah. So thanks for the question. Okay, then let's get started with the exercise 3, and I let Romy take over again.